Hello, everybody, to today's lecture. My name is Martin Schwarz. I'm the head of the Functional Neuroconnectomics group here in Bonn. Our group is embedded within the Institute of Experimental Epileptology and Cognition Research. So today's lecture is split in two parts. I will give a brief introduction into the main interests of the group. And in the second part of the presentation, Jens Schweihofer, a postdoc in the lab, will introduce you the topic, or specifically the topic of neuronal activity, behavior, and AI-based activity detection systems. So, Functional Neuroconnectomics group, the general interest of the group is the structural identification and also a functional characterization of neuronal networks in general within the mammalian brain, but we're focusing on the mouse brain, and the research is based on two main pillars. So on the one hand, we're interested in scientific questions, which I will come to in a second, but we're also spending a lot of time into on technology development. And on the next slide, I will briefly introduce you the scientific interest. So scientifically, we are focusing mainly preliminary on a functional characterization of the neuronal circuits involved into olfactory-driven behaviors. But we're also interested in circuit alterations associated with epileptic seizures. And lately, we became interested in assessing the circuit integration of programmed human graphs within resident mouse neurons. And this is in close collaboration with Oliver Brüstler here in Bonn. So some words to the technology development. So why are we doing technology development? We think that tools are very, very important drivers of scientific progress. And these tools are very necessary to uncover novel conditions. So the tools we are emphasizing on, that we are developing, are split. There's basically three types of tools. First of all, is viral-based tools, so viral-mediated gene transfer. We started 10 or 15 years ago. So we're using either AAVs, so recombinant adeno-associated viruses, lentiviruses, and modified rabies viruses to study circuit components within the mouse system. We are also working on light microscopic circuit analysis, and this is in close collaboration with Uli Kubicek here at the university. And light microscopically, we are focusing on light sheet fluorescent microscopy, and lately we developed light sheet fluorescent expansion microscopy that allows you to look at large tissue ensembles in great details in super resolution. And we're also developing several tools for tissue clearing. Since 2 years or so, with the start of Jens Schweihofer here in the lab, we are focusing on AI-based behavioral analysis, and we are basically using DLStream, and we developed a tool that is called DLStream, and Jens will give you more details on this. So on the next slide, now let me briefly introduce the topic of the lecture to you again. So if we're talking about linking neuronal activity to behavior, we need certain prerequisites. We need these prerequisites to understand the neural computations underlying these complex behaviors. And what we need for an understanding is we need to define a behavior. So we need to define the behavior, dissect the behavior into its subcomponents, and the subcomponents of a behavior we dissect into sequences of postures. Now, having this block of behavior specified, the second prerequisite to understand this, we need the structure of the circuits involved into this behavior. And here I'm talking especially about interconnected brain regions on a mesoscopic scale. So we need to identify the regions that are active during the execution of this behavior. And finally, and most importantly, what we need to identify the active circuit components for this specific behavior, the so-called behavioral engram. In order to understand the neural computations underlying the complex behaviors, what we need most importantly is we need to show a causal relationship between the active circuit components and the execution of this behavior. So in general, this can be achieved when you can demonstrate that an ensemble that is active during the behavioral execution can be affected can affect this behavior upon, and this is very important, upon instant interrogation. And we have developed such a system, which is called DLStream, that can achieve this. And Jens Schweihoff, now in the second part of the presentation, will introduce you to our approach. Thank you very much, and have fun during Jens' presentation.
Thank you, Martin. So let me introduce to you some key concepts that stand behind this AI-based behavior analysis. The first one, and I'd say the most fundamental, is so-called AI-based or deep neural network-based pose estimation. Neural networks are a type of machine learning algorithm that are inherently able to learn key features given from input data and then kind of translate them into a machine learning algorithm that can find these features again and again. Especially these convolutional neural networks have revolutionized motion tracking or pose estimation to a point where we are now able, as researchers with a very limited or very minimal data set, to define key points of interest and then go forward with markerless pose estimation. I'll show you some examples here that show you the strength and the capabilities of pose estimation as of now. On the top left, for example, you see a reaching task in rats where the joints and fingertips of the rat as well as the object are motion tracked or pose estimated in every frame. While in B, you can see a full body side view tracking of a horse going through the frame with the handler. And in C and D, you can even see social behavior of two different species. And previously, any annotation of behavior was done manually, meaning that a human rater would go through frame by frame videos and look and decide whether the behavior of interest is observed or not. This means that it is a very time-consuming task with very low level of detail, a high bias between raters, and especially rater fatigue happening, meaning that the same level of detail declines over a longer period of time. Now that we have this fast, reliable pose estimation, we can catch up with the high spatiotemporal resolution that is needed to look at neuronal changes or neural activity and decide, in this case, for every frame, what the animal is doing in fine detail. However, this is obviously just pose estimation, meaning to get behavior classification, we need a further step. So machine learning based behavior classification takes pose estimation as an input and provides a fast, unbiased classification in contrast to the slow and biased classification a human can provide. And another really great thing about this behavior classification is that because it is a statistical tool, meaning that it looks for patterns, for similarities, and for likeness and unlikeness in the data, using the right technique, we are able to uncover the behavior repertoire as a whole and look at the different nuances or sub-actions a behavior might have. This enables us to get new insights into the patterns and dynamics in behavior. Two of these examples of a representation of these statistical findings, this machine learning-based analysis, you can see on the left. Let's start with B. This is a two-dimensional representation of the behavior repertoire, ethogram, or map, whatever you want to call it, of an animal that was recorded from below. In this case, the machine learning algorithm was able to map the behavior and the sub-actions of these behaviors into a map, and then decide with the clustering algorithm, meaning that it looks for groups within this data, which ones are familiar, so which one belong to the same stereotype, and which don't. In A, you can see a hierarchical representation of that same behavior map you see below, where a human rater then, in the last step, looks at these clusters and gives them a stereotypic name that we would, or a definition that is very close to what we would use as human annotators when manually labeling. But what you can also see is, because this is a hierarchical representation, we are able to look at subgroups and their familiarity towards each other. So for example, in rearing, you see there are a lot of different subgroups, while in grooming behavior, there are little. And the similarity or the differences between those, so how far or how close they are related, can be found out by their similarity in this behavior repertoire, or by this similarity in this behavioral map. And taking this into account, we already have a tool that gives us a much finer, detailed analysis of behavior without even looking at the neural activity that might come into the same in the same timeline. So let's talk a little bit more about behavior classification and the different techniques that we can use, because there are two fundamental differences, and both of them have a good use for behavior scientists, and in the field of neuroethology, they have two. First, let's look at the easier version, supervised classification. So the step-by-step guide to supervised classification is pretty easy to understand. First, we need to identify a behavior of interest. On the right, you will see some resident intruder paradigms where we are looking at attack behavior and a lateral threat approach, for example. Then, as a second, you need to collect and label examples. This is similar to what I said about manual annotation, but here we just do it once. We generate a training set so that the machine learning algorithm understands what we as annotators want, because obviously the input needs to match the output. We need to be able to have a reliable classification that fits to our needs. The third part is that we take the post-estimation of these recordings, we extract the relevant features, usually something that happens within the technique. We train that machine learning classifier on the labeled data set so it knows what features are present whenever or change whenever the animal is showing that behavior. And then as a fifth step, we evaluate the classifier. Because we have an annotated data set, we can just see how well the accuracy of our classifier is in prediction. And then the last step, and that's the most important, we can detect the trained behavior in any video recording that we provide, and thereby have a fast and reliable behavior classification. Two examples you can see on the right, as I said before, with several plots where you have the behavior parameters as well as the behavior of the animals in a time plot or in a time series, and then you can look at the, let's call it, individual ethogram of each session, and if you change something in the session, you will directly see whether the behavior or the frequency of behavior that you're interested in is changed. The key advantage here, as I said, is you get what you label, meaning that by giving it a trained data set, you can define the behavior and also the nuance of the behavior that you're interested in. The limitation here is you only get what you label, meaning that what you have been showing the classifier is what it will show you. It will not show you any new patterns or new insights into the behavior repertoire. It will only give you what you wanted. This is a perfect chance to talk about the second kind of classification technique that actually gives us a more insight. Unsupervised classification is where, in contrast to supervised classification, where we'll take labels and teach the algorithm what we want, we look at the raw data, so in this case the post estimation, and then obviously the extracted features, and give the algorithm the opportunity to find their own groupings within. Something that you might be familiar from an earlier slide is this behavior map, right? So in this general approach, what's happening is that we look at the behavior of an animal, and then we find the statistical anomalies or the patterns in the data, and find these groups that are automatically sorted by the algorithm, but the algorithm has no prior knowledge to our final term or our final stereotypic behavior that we address to it. It is just looking at the data and the likeliness or unlikeliness of data in this high-dimensional space. So the steps are very easy. We observe the behavior, meaning we record videos, we post-estimate it. The algorithm then does unsupervised clustering, meaning that it finds the groupings We can then find the behavior map within, so the categories and subgroups within, identify common labels, meaning that we are looking at the groups and we decide what common stereotype they would incorporate. And then as the last step, we can look at the hierarchical representation, as we discussed before, where we look at the subgroupings. So these subgroups here, for example, and then either split them even further or merge them together in a bigger, let's call it family of stereotypic behavior. Now that I gave you a brief introduction into AI-based behavior classification and the two types, supervised and unsupervised classification, I think it is time to talk a little bit more about the behavioral relevance, or the experimental relevance in this case. So given the two types, we are now able to understand, or no, sorry, to identify behavior stereotypes that we were previously only able to annotate manually in post hoc, so in offline analysis. But if we really want to have a behavior dependent experiment, meaning that we react to the behavior online, give closed loop feedback on it, we need this ability to work online, as I said before. So this is why we started developing a tool that Martin already briefly mentioned, DLStream, or DLStream for short, that utilizes this online pose estimation, so this pose estimation, and then connects it to behavior dependent triggers, for example, using a machine learning classifier, to then orchestrate behavioral experiments that are cut or designed in a way that the experimenter, especially neuroscientists, can then investigate the correlations between neural activity and behavior. So let me just first introduce you to the concept of online behavior-dependent experiments. You will see in a, let me see if I can pause this. No, I can't. In A, you see the behavior of an animal, very simple one. It is just locomotion, whether the animal is running or not. And whenever the animal is running, which is shown in the green text, you see that this blue LED is starting, so is on, and whenever the animal is not running, it is off. In C, coming to this because it's not stopping, you see whenever the animal, so this is social behavior, whenever one animal is approaching the anogenital region of the other animal, which is already a stereotypic behavior. It's called anogenital approach, who would guess. The LED, in this case on the top left, would turn on. Whenever it's not there, it would turn off or stay off. And in B, this is from a classical conditioning task where we would condition an animal to react to an odor stimulant by previously associating a visual stimulus with either negative or positive stimulus. The animal is shown whenever it's close to the Petri dish with the odor, and looking at the screen, it will be shown either a plus or an X on the screen. So taking this into account, DLStream, which is, by the way, an open source toolkit that we provide a lot of guides and tutorials for if you're interested in trying it out, or in your future, if you're doing anything with behavioral experiments and you need closed loop stimulation. For example, this here is a good example for optogenetic stimulation, which I will show you a real example also in the next slide. You're welcome to check it out or to contact us if you're interested in this. And our paper is also available open source if you're interested in reading a little bit more about the scientific background. So the experiments that we've did based on DLStream. So let me talk to you about this for a second again. So behavior dependent experiment, meaning we have a behavior that we're interested in. We have a classifier or a very good definition of the behavior and what's happening during that behavior, taking pose estimation. We design an experiment that takes this as a trigger and then activates or orchestrates a cascade or just a simple stimulation protocol. We can already see here that it might be something very easy as optogenetic stimulation whenever the animal is showing a specific behavior. We also see that we can have more fine-tuned, more complex protocols in place where the animal is actually at a certain location, looking in a certain direction, where it is in contact with another stimulus like an odor, and it is shown a video or in general conditioned in a certain way. And then third, we are also able to use this pose estimation in social tasks, so behaviors that are usually very complex to rate by a manual annotator, and these can be automated. And when we're talking about correlating this to neural activity, we always need reliable. We need the same onset to be sure that we catch the neural activity that we're interested in. One of those examples that we are using and we have used for a publication is what I would shortly translate into, or sorry, shorten as translating neural activity into gene expression. We are using a light-induced activity-dependent labeling system called CalLight to, in this case, label active neurons that are active during a specific head direction. And again, let's first introduce the experimental design. You saw this already in the last slide. We have an animal showing a specific behavior. In this case, it looks in a 60-degree window that it can't see. And whenever it's looking into this, it gets an optogenetic stimulation in a brain region that is known for its head direction specificity. These animals were previously injected with a CalLight labeling system, which enables a light-induced activity-dependent labeling, and light-induced meaning that we connect light stimulation with the behavior and thereby translate or advance this labeling tool into a behavior-dependent activity, a behavior-induced activity-dependent labeling tool. This works in quite a straightforward way. We have one biological or biomolecular component that reacts to light and enables the next step, and the other one reacts to calcium rise and enables the next step. And if both come together, we have a gene, a reported gene expression. In our case, it's EGFP, but it can also be channelrhodopsin or halorhodopsin if you're then in a subsequent experiment are interested in stimulating the same ensemble or the same active neuronal population that you've labeled previously. If one or the other is happening, meaning if there is a stimulation, so a light stimulation but no activity, there will be no labeling, and if there is activity but no light stimulation, there will be no labeling. So this is kind of a key experiment that we've used to test our tool for the ability to correlate activity and behavior, because here what we can actually do is we can turn a behavior dependent light stimulation into an ensemble, a behavior dependent ensemble that we then as the next step can image and identify to get kind of a functional connectome or a behavior connectivity map, let's say, of our behavior of interest. So we can identify specific functional architectures that are relevant for that behavior by using these two techniques. But obviously, another way of doing this, if we are looking at, if you want to investigate neural activity, is to simply take something like channelrhodopsin or halorhodopsin, and then change the activity, the neural activity that we might simultaneously record with a fibroscope, for example, so calcium imaging, or with an electrode, and then change that behavior, change that activity based on behavior, and thereby investigating the correlation of certain neuronal populations to that behavior that we're interested in, and doing this online meaning that that we actually can look at the causality rather than just exploring the responsible regions by using something like large-scale changes like lesions or pharmacological inhibition. So let me just quickly summarize to get back to the points Martin made. First, behavior, meaning that we are now able to use closed-loop real-time behavior-dependent experiments, which is a huge step in the right direction. If we want to look at the neural activity during specific behaviors, we want to lock in these behavioral events and look at the connectome, or the let's say the functional architecture that is connected to this. So look at the causal links between those two very differently timed in the sense of on a very different time scale happening events. The key advantage here being coming from post-estimation, that we can look at unconstrained behavior, while previously most of the experiments in these kinds of directions were constrained experiments where the animal was either head fixed or the paradigm was so constrained that the animal could only do one specific thing in this task. So the analysis and thereby the correlation to neural activity is very straightforward. This is obviously another thing that I mentioned in post-estimation is that if you want to revisit certain aspects of these video recordings, then obviously not anymore in closed loop, you're free to do so, because this markerless tracking is able to or is backwards compatible, meaning that you can revisit old data, label new key points, and then do a more advanced, more complex analysis of these behaviors if you previously in your closed loop experiment didn't need to do this. The circuit architecture involved in this behavior can be investigated by using systems like CalLight, where we are now with tools like DLStream are able to do a behavior-dependent labeling of active neurons. And the third step, identifying the active circuit components. We labeled them in this last step, but using techniques like light sheet fluorescent expansion microscopy, where we can look at large-scale connectomics of labeled neurons, we can use these types of super resolution or virtual super resolution imaging to identify these functional connectomics on a larger scale, even connections between brain regions, long range projections, and so on, and then identify these networks and investigate them even further in more specific experiments if interested. One of the key limitations that I've previously talked about as well is the difference between timescales, the temporal resolution, so to say. Behavior usually is happening within hundreds of milliseconds and seconds, or even hours or minutes, depending on what type of stereotypic behavior you're looking at. But the neuronal timescale, so what the activity is happening at a millisecond timescale. So to connect these two, we need a very fine detailed, very reliable, very consequent detection of behavior. And one of the limitations of the system, at least the current state of the system, is that we can only react to behavior, to the behavior onset. So the moment we define the behavior happening, we are already reacting to the behavior. We're already lacking this maybe 2 or 3 frames. So let's say like this 100 milliseconds behind when we need to find out that it's this behavior. So in a real time behavior dependent experiment, the optimal time point would be to exactly react in that moment, or to stimulate in that moment when the behavior is happening, and previously knowing this. And this is one of the main features that we are looking at and developing right now, which I would call prediction, meaning that we move from the reactive behavior classification, where we have the behavior happening and then we decide, okay, this was the behavior that we're interested in, to a predictive behavior classification, where we look at certain markers or key features that happen before that behavior of interest, and then predict the onset of the behavior prior to its actual onset, and then start with the stimulation or with the manipulation of neuron activity exactly at the same time point as the behavior is happening. And with the summary, I would like to thank you for your attention. I'd also like to point out again that this is an open source tool, meaning that we have an open source paper and we have the code online if you're interested in trying it out or learning more about it, either now or in the future. And I'd like to thank you again for your attention. This was a lecture by the Functional Neuroconnectomics group. Thank you for joining us and have a nice day.